So, good evening from Israel. We're gonna do today a live scanning of a sample that I took some days ago from an urban pond in Tel Aviv. We got many urban ponds around the city with a lot of ducks and water lilies, and they house a large diversity of microbes and other creatures. And today we're gonna explore some of them. Okay, here is a picture of the sample I've took. Just some water without a plankton net. I don't like plankton nets because I think I get much more diversity when I take raw water. And with the water, I took some water lily leaves, which houses a lot of creatures that we'll see. We're gonna use today the inverted microscope, which gives us great flexibility. We just put a little bit of water from that jar on an open petri dish, and we can manipulate them during the process and switch to very diverse illumination techniques. We start with the 4x objective, so we can get a very broad view of our sample. We're scanning a sample of about 5 on 5 centimeters, and right now with 4x our field of view is 6 millimeters. So whatever screen you're watching, whether it be a smartphone or a desktop a computer screen or a large TV, the width of the frame now is equal to 6 millimeters. And I'm using now a technique, an illumination technique called dark field, which gives us a very nice contrast of the water and the microbes with it. So let's start methodically scanning from right to left and see which creatures we identify first, the big ones. And right at the beginning we can spot here two large hydras. And hydras are relatives of medusas, of jellyfish. They belong to the same class. They both possess nematocysts, which are venom cells on their tentacles. We can see it comprises of a large body that contracts and a number of tentacles. Here is one that spreads. These one are green hydras that harbor algae inside them that gives it its green color. We'll see all of these creatures in great detail after we finish the initial scan with the small magnification. Here we can see in the middle a Eucalyptus, which is a ciliate, a single cell organism. Here is another ciliate, which is from the genus Litonitis. Don't worry about the names, they're not important. You can just admire those creatures. Here is another ciliate called a stentor, and a stentor in Greek means a trumpet. You can see its body resembles a trumpet. Here's the diatomat. Diatoms are algae that surround themselves with a glass protective layer. Here is another stentor, and this one is a rotifer, which is a multicellular creature. Here's another ciliate, probably frontonid, we'll see it later. We can see this green bar in the middle, it's euglinid. Again, we'll see all those creatures in detail very soon. Here is another ciliate here. Don't know exactly who it is yet. And here is another beautiful rotifer, Testudinella. Moving on, another hydra. I'm just creating a virtual map in my head. Mesodinium, I suppose, a ciliate. Here's another ciliate, I don't know who it is at the moment. But it's good to create this map so we can go back to those creatures later in great detail. The big one here is a worm. We can see another Euplotus here, frontonid. Here is another stentor from a different species. Here's another stentor, Lytonatus. This is a testate amoeba called Archila. This one, I think it's Trachylomonas, an algae. We'll explore it later. Another stentor. Here is another hydra. It's in the process of the so-called walking. They can walk and hop. Here is another rotifer. All the squiggly lines that you see are probably cyanobacteria. We'll see it later. Another hydra. Another Archila testate amoeba, and a rotifer besides it. Here is another rotifer. Another hydra. Lytonatus, both are ciliates. A rotifer. This one, it moves like a worm, we'll see it later. Here is another rotifer in the middle. Another rotifer that travels. A stentor. Hydra. Looks like it's in the middle of catching a prayer. I think it's Trachilius, a big ciliate, we'll see it soon. And this is another hydra, a brown hydra, that doesn't have the commensal algae. Here is a very nice ciliate besides it, Loxophyllum. When I say ciliate, it's those creatures that bear cilia. Here's another one of those, Loxophyllums. Lytonatus, Loxophyllum. Here's an Hypatrich, also a ciliate from a different class. This one is a rotifer. Archila. some algae with rotifers around it. This one looks like a community of rotifers. That's something we should check. This one is a nematode. It's a worm, and it has a very peculiar movement. I think we'll start with the brown amoeba I've seen here somewhere, because I think it had a prey inside it. It was in the middle of catching a prey. So let's find it. Yep, here it is. Ah, it got loose. Good for it. Good for it. This one is also empty. Empty. Here is one. So I'm switching now for a different illumination technique called DIC, which will give us more detail and resolution, and we can see, yeah, that's a Trachelius. It's a ciliate. A very large one, like a floating balloon, and you can see it has like a head that protrudes. Here you can see it. Yeah. It's a predator ciliate, and often you can see it engulfing large prey. And here is Eupleutis, another very common ciliate. It's very fast, but we can track it. Right now I'm in the middle of the water column. Water column is about 0.5 millimeter. And now we see the tentacles of one of the green hydras, and you can see some lumps. Oh, it contracted, and those lumps contained the nematocysts, which are cells with like syringes with venom. We'll watch it more closely later with the 40x magnification objective, and now I'm in 20x. So the field of view is around 1 millimeter in the width of the frame, and we can see here on floating on the top of the water communal algae called Cyanura. Each bouquet contains a lot of single cell algae that lives together in a commune. We'll watch it more closely with the 40x. Here, you can see right now with more details the spikes that transfer the venom from the hydra's tentacles like a cacti. Not all the creatures are affected by this venom. Hydras are somewhat selective. I've noticed that they like rotifers the most. And now I'm going to the bottom, and we can see beneath the hydra body a single cell ciliate called Loxophyllum. Those warts, you can see the cilia around it, and those warts contains some venom organelles exactly like the Hydra, called nematocysts. We found another one that the Hydra doesn't mask, and the Hydra is just over it. And at the base of the Hydra there is a suction disc which enables it to anchor itself to various surfaces. Okay, now I'm back on the Petri dish button. You can see some scratches. Okay, here is that Laxophyllum. We'll go methodically. Here is again the Trochilius. It goes up and down, so it's not easy to focus on it. And here are very small ciliates from the Certophorid class. We'll watch it later in more detail with the 40. And here is one of our beautiful heroes, the Stentor. Again, the Stentor is a ciliate. You can see a lot of cilia, like eyelashes, that suck spray into his mouth. And those lumps inside are actually its nuclei. Let's continue. I'm going methodically. Here is another Stentor. This one, I think, perhaps it's in the middle of division, of reproduction. I can't be sure, but perhaps. Hydra. Sometimes there are even ciliates that lives on the Hydra tentacles. The ciliates are Trichodina, and they leave a commensal sharing, but not here. Okay, we've seen here another beautiful cilia. This one's called Litonitis. I think it's time to switch to the 40x so we can get more detail. And I need to just a second and adjust the color on my objective to fight spherical aberration. And here is Litonitis, and you can spot in its middle two large macronuclei, and there's a smaller one inside. Ciliates bears two kinds of nuclei, a macronuclei and a micronuclei. That's one of its characteristics. Beautiful Litonitis. Okay, let's get back to, let's say, 10x and see which other nice creatures we can explore. The biggest magnification is not always the best one, and for scanning and finding things it's much better to first use a smaller magnification and then go higher. And here above the Hydra we can spot an amoeba. So I'm switching back to the 40, and yes, that's a very nice amoeba. It's moving very slow, but you can see the flow of particles inside it. Actually, if we'll move to a different illumination technique called phase contrast, we can see it even in greater detail. And you can see the nice flow of particles inside it. Okay, back to 10x. With that, that's the Studinella. It's a rotifer. Always reminds me of the Starship Enterprise. You can see it now from the side. It has like a toe beneath it, and it's like a flat disc that hovers around. Yeah, we'll find another one later which gives us a better view, and right now it's on the side. Oh yeah, it flipped and we can see it's back. Watch the two manifolds on its posterior, sorry, the anterior. Okay, let's take a look at this nice worm. Going back to 20x, so we can estimate this worm to be around 1 millimeter, and it's surrounding another amoeba called Archila, which has a test, a protective home if you like. You can see the inner gastro tract of this nice worm. And who else we got here? There is a Trachelius hidden here inside the debris. Hydra, and this is a very nice stentor. All those green blobs that you see inside are actually food particles. Let's see it in more detail. I have to adjust the color of the lens. Yeah, that's better. Now you can see its cilia. Yep. Yeah, it contracted inside the debris. Okay, we'll find some more. Okay, so if we're here is a nice diatom mat. Diatoms I'm recalling are algae, and those green spots in the middle are chloroplasts that house chlorophyll to do photosynthesis, and they are surrounded with a glass protection layer, and around it we can see a Trachelius, again a very large, magnificent predatory ciliate, which is now very high in the water column, so it's a problem to get a good view of it. Yeah, now it's fine. And here is another datum, a lonely one. Here is another Archila. The difference in color, it's due to its age. Older ones are absorbing metal depositions from the water, so they get dark and orangey, like rust. And right beside it, we can see a little euglenid, which is an algae, and that red spot that we can observe, it's so-called eye or stomata. It's not developed eye, it only helps it to discern luminosity levels, black and dark, black and white. Let's go back to 10. Another stentor, diatom, stentor. I think we got to the starting of our sample, yeah. And here we see a hypotrich. It's a ciliate. Let's watch it more closely. Yeah, that looks fine. And you can see its cilia are somewhat thicker than what we've seen before with other ciliates, and that's because hypertrichs have like merged cilia, that helps them to kind of walk on surfaces. I need to adjust the objective better, but this guy doesn't want to stop. Oh, now it's much nicer. And you can see those thick cilia. Okay, back to 10. I'm reminding you, if you got any questions, please write it on the chat. I soon answer them. Here is again the Tlaxophyllum, a ciliate. Hydra, rotifer, Certophorid, ciliate. Again, that hypotrich. Let's go to the upper part of the sample we haven't explored yet. This one I suspect was a snail. And here is a water flea. So neat. Water fleas are actually small crustaceans. It's very thick. You can see all its internal organs inside. Just let me adjust the illumination here. Let's go back to 10. Here is another rotifer. And here is a nice euglanid that wants dwarf that will enlarge. Okay, this one resembles like a green candy snake, and you can see this red spot in the middle, its eye, stomata, and it's actually an algae. You can see in its middle rectangular shapes, which are deposits of starch that it preserves for later use. It's called paramelion. It's a kind of starch compound. What happened here? That's better. Let's get back to 10. Okay, here's a nice rotifer. Again, rotifer are multicellular creatures. You can see like a red spot on its back. It's actually its eye. This one has an egg inside it. And here we can see another ciliate from the Paratrich family. What's nice about viewing a sample in a Petri dish is that we can see two neighborhoods actually. The bottom one, where the creature likes to crawl on the surface, and the top one, which we'll explore later, which contains other organisms that lives on the biofilm of bacteria that floats around. Here is another rotifer. Here is another stentor beside the hydra, and pseudophorid ciliates around it. Okay, this ciliate, I actually don't know what it is. Could be some type of colpoda maybe, but I'm not sure. I think it's a colpodid. Maybe I'll take it for greater examination later in another microscope. But back to where it came from, we can see a mosquito larvae. I think it's midge larvae. Tentacles of hydra. Let's go back to the bottom. I moved two down. Sorry for the fast movement, I'm in the middle of scanning. Actually, I'm doing it much slower than I'm used to. Here is again the Trachelius, magnificent predatory ciliate, like a big balloon. Oh, here is a nice one. Go to the 40 to see that one, and this one is an amoeba. Its former name is Hyalodiscus. I don't remember its current name, but this amoeba belongs to the vampirilids, which are amoeba that specializes in attacking algae and sucking them like a vampire. Okay, let's go back to 10. Here are two rotifers. Oxophyllum, hydra. This is just a small sample from that pond. It's actually one of the first ponds I was taking samples from. but the sample is a few days old now. Oh, here is a nice ciliate, Mesodinium or Strombolidium. I can't remember the difference, sorry. One of them, oh, it jumped. Yeah, those are very fast. Like a strawberry. Yeah, no chance to finding it now. Let's see if we can spot another thing that we haven't talked about. Maybe Trachelamonas that I've seen earlier. And then we'll move to the top layer, the bacterial layer. That's a worm. Euplotus. Watch how it uses its cilia-like legs to climb on things. Rotifer. Here is another one of those amoeba that we've seen later. I want to look at it with phase contrast. I think it got a prey next to it. So let's explore it with the 40x. Okay, nice. So with phase contrast we can see the orange body, and on the bottom of it we can see the pseudopodia, the so-called leg that the amoeba uses to move around and stretch. And it surely caught at something. It's in the middle of digestion, I think, some kind of algae, but I can't be sure who exactly. Okay, back to 10. Stentor, Hydra. Hydra. Oh, two very nice Loxophyllum. Let's look at them in 40x. They're worth it. Magnificent cilia. I'll just adjust the objective a bit. Yes, it's a very nice view of it. You can see the fine lines surrounding it, which contains the venom cells. Supposed to be another one on the right. Here it is. You see they change shape quite often, from elongated to more dense one. And it has its nuclei scattered all around its body. On the left side you can see those lumps are what called warts, one of the characteristics of this genus. back to 10. Enter. Rotifer. Who is moving in? Oh, the nematode, the worm that we've seen earlier. Here is another Lytonatus. Let's see if there's something on the water border, the sample border. Here is again that Strombolidium hovering like a little strawberry. And I can't find the Tricholomonas that I spotted earlier. Oh, here is another small ciliate, very common, called Halteria, and we can ID it from the way it moves and jumps around. You see it's very, very peculiar behavior. Here is another one called, I think it's a frontonid. Let's look at it in more detail. Yeah. Downsides of using a petri dish, it's the water column is not even, so creatures can go up and down. Stentor, you can see how it sucks particles into his mouth. This one is, I think, a zoospore of an algae. One of the ways that algae are reproducing, it's sending zoospores. Here is that Alteria we've talked about before. A Paratrich ciliate next to this worm. This one is dying. You can see the bloating on its left side. So let's move up to the top layer of water. And switch to phase contrast. Let me adjust the ring. And now what we are seeing, those spots, are bacteria. Bacteria that floats on the top surface of the water. And we'll sure find some creatures that harbor it soon. Here is one, yeah, a certophorid. See running on the top layer of water, harboring those bacteria. Also, a lot of flagellates and amoebas like to be on that layer. But I don't see a lot of them right now. Here is the Halteria that we've seen before. See, it just goes up and down on the water column, so it's very difficult to focus on it. We should stay at one spot till it comes back. Sometimes it stops. But it's a very active one. Ooh. I think that Euplotis got strung in something. And all those fine strands that we see scattered around are cyanobacteria, which is kind of a mix between bacteria and algae. Let's see what else we can spot with phase contrast. The Hydra. The width of frame now is 0.5 millimeter across. Certophorid. And I can't find those nice Trochelomonas, so I think that's enough for today. We've seen a lot of species, and next time that I'll find an interesting sample, I'll surely, just as I was talking, here is Trochelomonas, so I'll move back to the IC, and we can look at it in more detail. Trochelomonas is a very nice algae that covered in a protective lorica or shell. I'll add some more magnification and adjust the objective, and we can see that it has a red spot, its eye inside. The protective house is kind of spiny, and at the anterior part of it we can barely spot a flagellum, a single flagellum that this algae is using to catch prey and for locomotion. And a very small heliozoan here as well, called a sun animal, heliozoan, because it resembles the sun with its rays around it. So I think that's enough for today. Have a great evening wherever you are, and I'll see you again when I'll have a nice sample to scan together. Bye bye.